Hi, we just gathered here to make the initial planning and choose the PhD study sites we need to work on. As our primary objective on finding study site was selecting landscapes with rubber, tea and open area land use types, we need to visit several estates and forest reserves to inspect the suitability of the study site. Since we got a permission delaying issue from the forest department at the initial stage of the PhD research, I must thank and acknowledge the Canon Valley plantation of Haley's group letting me work on their estates. So we visited Halgol and Vioya plantations and selected several land blocks to carry out our research. Basically, we have two treatments in my experiment as disturbed landscapes and undisturbed landscapes. In this map, you can see those two different categories where disturbed landscapes denoted by red color, basically the plantation sites, and undisturbed sites denoted by green color. These sites were the forest reserves or protected areas. One of the most challenging part was finding a proper landscape blocks to match with my experimental needs, where I need to have different land use types within the same local setup. Therefore, we need to walk and observe the land before we implement the project. It took me several months to finalize the study sites in the disturbed landscape, as we need to concern about many factors including transportation, logistics, and our target bird species. My team need to communicate immensely with estate managers, staff members, and also with the local people in order to get a clear picture about the working field, because the local people have their grounds and also they know their grounds better than anybody else. So I always encourage every researcher should respect and get the necessary assistance from the local people as much as possible. So this is Halgol Estate, where we have selected it as the second study site after Viola, which has tea plantations as the dominant land use type. It also has small portion of rubber, secondary forest patches, human settlements, roads, and open areas such as grasslands. We had a special interest on this type of abandoned secondary forest patches, as these vegetation patches could hold at least a few species of forest birds, because we need to find one of our target species, the Sri Lankan Brown-cappedBabbler, an understory forest specialist. This is a wide-angle view of typical landscape cross-section that we have to consider when conducting the bird translocation experiment, because we have to give the opportunity to the bird to select its preferred land use types to move through. Then we have to select another study site from Kaltura district due to COVID travel restriction between districts. So here we selected a mixed culture estate located in Kudaligama to continue the research works, which also had rubber and tea plantations, as well as Tickel'sBlueFlycatcher and Sri Lanka Brown-cappedBabbler. Before we implement the translocation task, we need to locate the focal individual, then get the information on that location, such as vegetation condition, disturbance levels, and fragment size. This location is the capital location for the individual, and we need to determine the release location as well, which is also very important. So we need to be in the landscape and get a proper idea of all the land use types, because we also need to move through those land use types while tracking the birds. Believe me, it's not an easy task. Here we are in Sri Lanka, a small tropical island in the Indian Ocean, and we are here to do a special kind of avian experiment called bird translocation experiments. There are several translocation experiments like conservation-oriented translocation experiment, management-oriented translocations, and so on and so forth. But we are doing experimental translocations, specifically concentrated on landscape ecology. Basically, we want to know how birds going to use a particular given landscape in their day-to-day movements. For example, you can see in the background, you can see rubber matrix, and also within this rubber matrix, you can see isolated secondary scrublands, secondary forest patches, which is like green island in the rubber ocean. So we still don't know how those bird species going to use these different matrices apart from their natural habitat. So the plan is we're going to catch bird species from a big forest patch, so we call it a capture site, and we translocate that individual bird into a different matrix which has some sort of cover, initial cover, as a release patch. Then we, before we release the bird, we mark the bird with a ring, as well as we put radio transmitter which emit a specific radio frequency, so we can locate the bird, or we can locate the exact position of the bird with a radio signal receiver and a Yagi antenna. And then we can track fine scale movement of that particular bird species in order to get the clear idea of how that bird going to use the specific land use types or land matrix, even the landscape elements such as isolated trees, corridors, small forest fragments, and so on and so forth. So far we have translocated few individuals of Brown-cappedBabbler and Tickel'sBlueFlycatcher. So we use those two species because of a specific reason. Basically, Tickel'sBlueFlycatcher is a generalist species where it uses many wide range of habitats like human modified landscape, forest patches, plantations, and many. We can see Tickel'sBlueFlycatcher within many habitats, but Brown-cappedBabbler is kind of a habitat specialist, so you cannot find Brown-cappedBabbler in disturbed habitat. Basically, for example, like this rubber matrix, you can't find Brown-cappedBabbler unless it has a thick cover of vegetation. So far there are more than 28 or 30, around that number, 28 experimental translocation studies in the world specifically target in landscape ecology to track fine scale movement, but there are no studies in the Oriental region. So this study is going to be the very first study which focus on Oriental species, and Brown-cappedBabbler is an endemic bird. In other words, it's a restricted range bird, and we hope to find interesting results from this study. Here we are in Halgolla plantation of Canon Valley Group. This is a study site belong to undisturbed landscape, elevated landscape about 600 to 1100 meters from sea level, with undulating terrain. So let's see how translocation works going to be here. So this is a secondary vegetation patch, but it's comparatively less disturbed. And when we play back the call of Brown-cappedBabbler, we got a response, and we have seen one individual within this patch, and we're going to catch that individual from this patch and release it over there, because in this landscape we have both tea plantation as well as some kind of open grassland area, so it may have some stimulus to home back to its capture location. So tomorrow we're gonna do that translocation experiment. Nearly it's like 200-meter distance from capture location to release site. Yeah, today we need to find a release location for the Brown-cappedBabbler. Behind this building we have a small forest patch where we're going to capture the bird, and we're going to translocate that bird within the 500-meter radius. So we need to find a release point where it has a internal stimulus to leave that place. Otherwise it may settle in that release patch. So we're gonna patrol the area to find a release patch for the Brown-cappedBabbler. Let's go and see. Once we select the capture location, we need to erect mist nets with least disturbance to the remaining vegetation in order to capture our target individual Brown-cappedBabbler. We need to use playbacks to attract the bird to the mist nets. Mist netting is a very sensitive technique where you need a high level of expertise to capture and handle the birds. Of course, you need to have permission from Department of Wildlife Conservation to capture birds, as well as to use playbacks. And also you need to take ethical clearance permission to handle birds and put transmitters on them. So we got all the necessary permissions before we start our fieldwork. It's a must to obey the fauna and flora protection ordinance when you do fieldwork in Sri Lanka. When birds get entangled in the net, we extract the bird carefully and put it into a bird bag until we process it. We need to collect biometric data and need to attach a radio transmitter before we release it to a release patch. Since capturing a target bird is so difficult, we need to be super cautious to record all the biometric and morphometric data when the bird's in our hand. This is the first Brown-cappedBabbler we're going to translocate. To study the moving path of this bird, we have attached a radio transmitter. You can see the aerial type of thing protruding the back of this bird. We use backpack harness method to fix the transmitter on the back, and also glued it with eyelash glue. After a few minutes, we're going to transport it to a release point away from his usual territory, and we will monitor its movements using radio biangulation, so we can understand how the land use type selection of Brown-cappedBabbler when it homing back to his own territory. Here we are in the release site, and you can see it's also a second vegetation patch or a small forest fragment where this individual can take the initial cover and rest to recover from our handling stress and transportation stress. And this vegetation patch also not enough to settle down, so it need to get away from the vegetation patch because of the small size, roughly less than 25 square meters. The surrounding area dominated by monoculture rubber plantations where it need to penetrate to go to the capture site. So let's release the guy and observe the initial behavioral response. It's a must to observe the individual bird after release because it was kept in a hand and the bird is now with a radio tag and a ring, which are both alien objects to the bird. So you can see it performing some preenings and conditioning the feathers before it ready to move into new locations. So it's going to do exploratory moves to know about the new habitat. Even though it pecked on the ring several times, the initial behaviors tells us it quite okay with radio tag and the ring. So we can begin our monitoring the movements of this bird using radio triangulation technique. Before that, let me introduce the technique we're going to use in the field to fix moving locations for these birds, which is known as radiotelemetry or radio triangulation.
Yeah, so now we are going to do radio telemetry, which is we put transmitter on a bird, and that radio transmitter emits some specific frequency radio signal, and this Yagi antenna and this radio signal receiver. We going to use these two equipments to get the correct direction. Actually, we need to collect at least 3 directions simultaneously, 3 bearing to get the location by doing radio triangulation. First of all, we need to adjust the signal gain and the tune in order to when we adjust the tune, we can listen, we can hear some kind of beep, which is the indicator of receiving signal, and then we need to keep the antenna by this handle. Never ever touch this metal part because it's gonna earth the signal, and it's going to decrease the performance of the signal collection. So always try to remember to hold the antenna by this plastic handle, and then you need to point it and gently rotate 360 degrees until you get strong beep signal. So we are getting signal from this direction, and by using a compass we can note down the direction where the signal comes, and quickly we need to change the location, or we can use another receiver to get the another direction, another bearing, and then if we take it like simultaneously like same time, you can use those 2 directions and you can get the crossing point as the location of the bird. This is Harsha here, taking a direction from a location. Here Indika here, taking another direction, another bearing from another location.
So we monitored each and every individual for 3 consecutive days until it homed back to its original capture location, or continued with monitoring up to the 3rd day. If we confirm that the individual bird is not going to leave the release patch, we decide to capture it again and return it to the on territory, and remove the radio tag or the transmitter. We usually collect location data once every 15 minutes to match with the average time intervals used by other studies done in other countries. Basically, from 6 a.m. to 6 p.m., or the from dusk and dawn, morning to evening, we continue this monitoring process, and continue the fixing location for once every 15 minutes. um, Here, the one we translocated at Halgol plantation did not return to his territory or capture location. That Brown-cappedBabbler showed kind of exploratory movements, and when it come to the tea plantation, it again bounced back to the uh, release fragment. So clearly showed reluctant to penetrate the metrics in between capture and release location. So we monitored it for 3 continuous days, but then we realized that uh, it's not going to move to hit its release location. So because of that, we decided to recapture it again using playback on the third day. But it did not respond to playbacks because it did not respond to the territorial call. So we were unable to capture it or attracted it to the mistnets. So we had to use a different strategy where we locate the roosting point thanks to the continuous radio signal during the night and captured it.
So we have a situation. So we translocate this guy Brown-cappedBabbler to this release patch, and we monitored it for 2 days. Seems like this guy is already established a new territory in this release patch. So we decided to recapture it again and release it into the original captured location. So we remain until night falls, and when this guy went to sleep, we find the location by radio triangulation, and with the use of net we captured the guy again. So now we're going to remove the transmitter from its body and release it to the same capture location tomorrow morning.
At the very beginning of the experiment, we got several failures. There was a one individual which dropped the transmitter by breaking the thread. So let's see how we found the lost transmitter.
19 November the 10th. So this is our fourth day of the experiment, and yesterday we got a heavy rain, and after 4 p.m. We tend to get a direction like same direction since 4 p.m. So we suspect whether the bird has removed the transmitter, or so we don't know. But today morning, yesterday we saw an individual without transmitter, so we suspect it probably could be our sampled individual. And now we are still getting a strong signal from a single location. It's not moving, so we suspect whether that the bird already removed the transmitter. So let's try to locate the transmitter position. So I'm going to lower down the power, so because we are getting very strong signal, and try to find the correct direction from here. So it's indicating this direction. And what about you, Indika?
Yeah, I also getting the strong signal from this direction.
So let's see. So we need to hypothetically mark the line, the direction from both antennas, and we need to find the exact location where those two lines meet or intersect. So we can imaginary get a location, get a position, and let's go and see, and then let's go and search that location to find the transmitter. signal getting very stronger. Even with the low again. Yeah. So the transmitter definitely fall down in this area. You cannot see any bird, so it's definitely the bird has removed the transmitter. Let's try to find. So it's this direction and this direction. You can clearly see the intersect where the two lines cross, and I can see the transmitter exactly the position where the two directions cross. So just remove the transmitter. You can see this is the radio transmitter, and it has cut the nylon thread. See, you can clearly see, and you can see that bird has tried to move to the edge of this vegetation patch, but it's never ever penetrate the rubber matrix. And probably the bird that we saw yesterday could be our target individual, and it has removed the transmitter. So now we try to capture it and release into the same capture point, but without transmitter it's so hard to find the bird, and we need to try a little bit hard to capture that individual. Yeah.
When it comes to capturing target birds, it's very important to know about their microhabitats, flight pathways, and behavior patterns. As a part of the same research, we had to study the habitat usage of both species. So we had to monitor this brown-cappedBabbler for several days to identify moving paths where we can put a mist net and capture it. Since it has lost its transmitter, we have to monitor it because we need to capture it, however. So we found it tend to cross this small stream several times. Thus we put a mist net across that flight path. Since the brown-cappedBabbler is an understory living small babbler, we don't need to elevate the mist net to higher levels as we did it for the Tickel'sBlueFlycatcher. As soon as we captured that individual babbler, we quickly release it to previously captured location and remove the mist net from here. Since we put the mist net across the small stream, accidentally we caught a forest jewel in our mist. One of the most beautiful forest dwelling kingfisher, the oriented dwarf kingfisher, which we'll show you in a shortly. We will show you it lately. So mist netting is a very time consuming technique where you need to do it very carefully. If you are interested about mist netting and if you want to engage in mist netting operations, you can join with us at Field Ornithology Group of Sri Lanka. It's an environmental organization established in University of Colombo Science Faculty, patroned by Emeritus Professor Sarath Kotagama, who was also my local supervisor for this PhD project. Field Ornithology Group of Sri Lanka, or well known as FOGSL, has beginners workshop to teach and train researchers, students and volunteers who like to actively engage in bird and wildlife conservation. So anybody can come and get the membership and join that program to get first-hand experiences in mist netting, bird handling, and even avian research techniques. And also FOGSL, we have collaborations with Department of Wildlife Conservation to conduct National Bird Ringing Program, where you can participate as a volunteer when you obtain the certificate from beginner's workshop. Okay, let's back to our translocation experiment. So we captured several individual brown-capped babblers in this whole experiment, both from secondary forest patches situated within the rubber estates and in natural large tract of forest or in protected areas. And we found that all the individual brown-capped babblers responded differently to playbacks, where some individuals aggressively came directly into the mistnet. Some individuals did not come to mistnet but called at a distance place, even though we have well camouflaged ourselves to hide, to reduce the observer presence or show the absence of observers. And some individuals never responded at all. All the individuals of brown-capped babblers we translocated within the plantation landscape setup never return it to capture location except for one individual. All the other individuals need to recapture and relocate to the original territory or the home range after 3 to 5 days of continuous monitoring. Those individuals, babblers, performed exploratory movements within the release patch, which always bouncing back to the same patch of vegetation once it penetrates into rubber or open area matrices. Even though this rubber plantation had some ground cover with some ground creepers, the brown-capped babbler only remained there for no more than an hour, but quickly returned to small forested scrubby patch when we have released it. This tells us the importance of having some sort of understory forest cover to facilitate the movement of brown-capped babbler, because from our observations we showed that it's looking for some sort of understory cover in order to perform its flight or in order to put it flight pathways. Here you can see the typical monoculture rubber plantation without understory vegetation, which might be a movement barrier for understory living birds. And this is our brown-capped babbler, which was failed to return to its capture location. So we have to recapture it again. So we have to actively flush the bird into the mistnet, as it did not cooperate with us for playbacks. So our team worked together in order to directed it towards the mistnet in order to capture it. So this brown-capped babbler, it was unable to orient toward its capture location. So this is our 5th day of tracking this bird, but this guy unable to move back to its capture location. Because of that, we're gonna remove our transmitter from its back, and we are going to release this guy again into the same capture location. So we have fixed the transmitter with use of eyelash glue. Because of that, we have to put some water to dissolve the eyelash glue and remove the transmitter from its back. Then try more water just to clean the remaining glue, and we have put a blue ring to individual identification, and get to the photographic grip and make sure it can flap. Yeah, so it's quite normal in behavior. Now we're going to release this guy into the same capture location, so it can flap, which means
it can perform its normal flight behavior. Let's release this guy into the forest patch. So this is the exact location where we put our mist net in order to capture this guy by playback, and now we're going to release it into the same location. So I'm going to release this guy to its home range, natural home range. Okay, here we go. Your natural home.
Let's see about Tickel'sBlueFlycatcher. Not as Brown-cappedBabbler, Tickel'sBlueFlycatcher was the main challenge when it comes to capturing the target individual, because it is a mid-canopy and canopy layer species, so we have to monitor its behavior immensely to identify the most suitable location to put our mist net. My field crew worked hard to find the movement pathways within the territory of this Tickel'sBlueFlycatcher, because it was not that aggressively responding to the territorial playbacks we played. So we have to continuously monitor the focal individual to find out the flight or the movement paths which the bird most preferred, as well as where we can erect our mist net. It took us more than 3 days to determine the netting location, because we need 100% success when it comes to capturing high-flying bird species. And Tickel'sBlueFlycatcher is a strong bird when it comes to the flight performance as compared it with the understory dwelling Brown-cappedBabbler. Furthermore, it was not that hard as in primary forest to capture Tickel'sBlueFlycatcher when we were trying to capture it from disturbed areas or within abandoned forest fragments situated within these estates, because the average vegetation height was no more than 15 meters. And the natural behavior of Tickel'sBlueFlycatcher made us to be super patient, because it is an insectivore where it sit and wait for long period to capture and flying insect prey, but quite active during dusk and dawn. So it's important to track the movement during morning and the evening to get a better understanding of the flight pathways. Thanks to the dedication, commitment, and patience of the research team, we were able to find few places where we can try mist netting for Tickel'sBlueFlycatcher. So we put our mist net and do the playbacks as well. After several days of trying and several few failures, we caught our target individual of Tickel'sBlueFlycatcher. As soon as we extract the bird from the mist net, we measure the biometric data and proceed to the uh, attachment of transmitter uh, to conduct our main objective.
So here we are in the capital location of this Tickel'sBlueFlycatcher. We put this net and played back the territorial song of this bird, Tickel'sBlueFlycatcher, and we caught this individual. And we already put red color ring, plastic ring, to individual identification, and we already put the radio transmitter. Here you can see the antenna of the transmitter, and we use backpack harness method to put the transmitter. And now we're gonna translocate this bird nearly 300 meter to a open landscape where, and we're going to track back the homing path of this individual. And this is the second Tickel'sBlueFlycatcher in this experiment. Let's see how it goes. Okay, now we are in the release site, and it's roughly 300-400 meter. Not sure about that. We can measure it later. And we're going to release this Tickel'sBlueFlycatcher into this small vegetation patch where it can has an initial cover to hide out. And let's see how it's behaved. And just last check whether it can fly. Yes, it can flap its wings. So we're gonna release this guy into this vegetation patch. Now it's 23 past 10. Let's see. Okay.
Not like Brown-cappedBabbler, tracking a Tickel'sBlueFlycatcher is very interesting because it moves fast from one location to another location, where Brown-cappedBabbler tend to hop on the ground with small distance steps and stay long time in one location. When talk about field equipments, we use TRX-1000 weather resistant signal receiver purchased from Wildlife Materials Incorporation in USA for radio signal collection, and BD2 radio transmitters from Hollow Hill Company as signal emitters. And also we use 3-element Yagi antennae to grab the signal. We recorded observers, locations, GPS data as latitude, longitude, elevation, and GPS signal error for both observers or both signal collectors, and wrote down the signal bearing direction using compass or smartphone compass app. Then we can use those data to calculate exact location using a special software called LOAS, which is a dedicated software application for radio triangulation or biangulation calculations. The software was developed by Ecological Software Solutions LLC, but only available for Windows operating systems. As we mentioned earlier, we noted all the GPS data once every 15 minutes, as it was the average time interval used by majority of the experimental translocation studies conducted in elsewhere in the world. Once we finished collecting data in the field, we used to transfer those field collected data into the proper data sheets. Then we can double check for errors which caused during the field note taking process. At least 2 people were actively engaged in checking the data uh, especially the GPS locations and signal direction angle for errors. Each and every latitude and longitude values, time, and signal directions were compared with a roughly sketched map before transferring field notes to the datasheets. We couldn't work at Viola and Halgolla area during the corona pandemic situation because of the travel restriction, and my supervisor, Professor Eben Goodell, asked me to be innovative and find creative solutions to such situations. So we had to look for a new study site within my home district. During corona pandemic lockdown, we worked at Kalutara district in my father's own state at Kudaligama. I was lucky enough to have a estate with both rubber and tea, as well as both of my target species in the same estate area. So, as usual, my research crew patrolled the entire area and found a territorial Tickel'sBlueFlycatcher, and early in the morning we went to put a mist net and capture the bird. When capturing the Tickel'sBlueFlycatcher, we had to increase the mist net pole height, so we used wooden sticks attached with aluminum cylinders to the other side of the stick to extend the mist net pole lengths. We had to walk roughly 500 or 600 meters to reach the forest fragment where we have observed the individual's Tickel'sBlueFlycatcher, and it was located just next to a rubber plantation. So we decided to put the net at the edge of the habitat and used playbacks to capture it. Once we captured the bird, we quickly removed the net and went back to process it and do the translocation. Another important part of this experiment was doing the control experiments where we need to capture the both species from natural and primary forest, basically within large tract of forests declared as protected areas. So we worked at Makandava, Bodhinagala and Yagirala forest reserves to conduct our control experiments. So we explored the area for our target bird species and marked the locations to capture the individual birds. Further, we did home range surveys as well to understand the habitat usage of these species. It was very obvious fact to us the individuals from protected areas use much larger ranges compared with the individuals from disturbed landscapes. Within the protected area, we started with Tickel'sBlueFlycatcher and we captured and ringed an individual. As usual, we collect all the biometric and morphometric data. Because we hypothesized the individuals in protected areas are much healthier than the individuals in disturbed areas, assuming the individuals in disturbed areas might have few or lower feeding opportunities. So we can use body measurements as a proxy to get an idea about the health condition of the bird. As we use body mass index, we are using among humans to see the overweight conditions. Not only that, basically every ornithologist used to take basic biometric data or basic measurements when we capture a bird, because we have to put huge effort to capture a bird, so we can use those data later on for any other research. We selected the dry season of the wet zone to capture the bird because it's easy to work within rainforest during the dry season. As you can see in the background, the forest floor was much drier with less damp condition. And I had to repeat again that you cannot capture and handle bird like this without the permission from the Department of Wildlife Conservation, as well as without expert knowledge on handling wild birds and handling measuring equipments. If somebody interested about the data, it is all available at Fogesel office at the University of Colombo or in the Wildlife Conservation Department head office under the research division.
Yeah, here we have captured our very first individual male of Tickel'sBlueFlycatcher from the low country rainforest. This is the first individual within the rainforest, because previously we captured Tickel'sBlueFlycatcher individual from rubber and tea estate. So this guy is the first one from the natural habitat, and it's been quite challenging to capture this guy because it's perceivably the home range size is quite large, so it's tough to identify locations where it fly in low levels, because in rainforest the average height it's about like 25 meters, and this guy is flying about 15 to 20 meter from the ground level. But we were able to observe a location where it comes very close to the ground, and we put our mist net at that point, and we were lucky enough to capture this guy. And we put red ring on the left leg. Now we are going to release this guy and continue to monitor the home range size of this individual from Makandawa Forest Reserve at Kitulgala. Let's see how it goes.
In our experiment, we used individuals marked with color rings as well as the individuals which tagged by radio transmitters in these home range surveys. When it comes to the individuals with rings and without radio tags, we need to actively engage in monitoring the individual bird. Thanks to territoriality of this species, we got a rough idea about the boundaries of the home range, and it is very important to monitor a bird as soon as we release after a handling session, because we don't know the stress condition of the bird while in the hand, and post-release behavior is crucial to record to make sure that bird is behaving normally.
Currently we are recording exact perching point of our ringed Tickel'sBlueFlycatcher to map its rough range. So it will be easier for us if we can mark main reference point on a rough map. So we are collecting possible information as much as possible to create rough sketch of map of Tickel'sBlueFlycatcher. For example, this type of tree we can identify it as a reference point because it's quite large in DBH value, and it's a Dipterocarp tree with nearly 40 meter height. So we use this type of landmarks to mark our initial map as reference point. So currently our team members involved in that process. Then when we do exact mapping thing, it will be easy for us to record exact points and exact reference point to create actual home range size of this natural habitat living Tickel'sBlueFlycatcher.
When it comes to active observation of birds in scientific studies, we have to think about observer influence, because as observers, our presence might affect on their behaviors. So we need to habituate them towards us, or we need to be well camouflaged to hide ourselves in the habitat. But in the case of Tickel'sBlueFlycatcher and Brown-cappedBabbler, they were quickly habituated to us, and we observed normal behavior even with the presence of our close proximity. We need at least 2 observers to collect perching points once every 15 minutes, where one observer was continuously monitoring the bird and other observer was recording the locations.
So we captured another Tickel'sBlueFlycatcher within the primary rainforest in Makandava forest at Ketulgala, and this is our second individual for the control experiments, where we capture the bird and translocate it within the forest. So basically we can compare the homing time and homing success between the two sites, between the two experiment setup within the primary rainforest and outside the forest, basically the plantation areas. So for the both species, Brown-cappedBabbler and the Tickel'sBlueFlycatcher, we have those control experiments, and this is the second individual for such control experiment for Tickel'sBlueFlycatcher. So we already put the transmitter and we put yellow ring to the right leg, so we can identify it individually even it lose the transmitter. So everything is ready, and it can perform the flight. So you can see the it can flap the wings, so it's quite comfortable, no stress behavior at all, quite energetic. So we're going to translocate this individual within the forest, within the selected distance, basically less than 500 meters. Let's see what will happen.
Then we move to another section of the rainforest to do the control experiment on Brown-cappedBabbler. So first we need to put mist net and capture it.
Okay, so we captured our third individual of Brown-cappedBabbler for the control experiment. Actually we captured both male and female, and we measured the female and release it in the site, and we translocate this male to a different location, approximately 350 meters. So now we are going to release this guy. We have put light blue ring to the right leg of this guy, this individual, and we put 202 frequency transmitter in order to get the homing back location. So let's see how it's home and how it's performed the homing success and homing time within the primary forest in this Makandava forest reserve as the control experiment. Okay, it's time to release.
We were quite amazed by this Brown-cappedBabbler because it did not move away from us but settled just next to us, maybe 3 meters away from us. After spending nearly 3 years of facing various challenges and obstacles, we were able to translocate 22 individuals, where 5 Brown-cappedBabblers in disturbed areas and 4 Brown-cappedBabblers in primary forest habitats. For the Tickel'sBlueFlycatcher, the total number was 10, where 7 in disturbed areas and 3 in controlled experiments, which is forest, primary forest. Therefore, altogether 19 successful translocations were done with 3 fails. We also completed 13 home range surveys for Brown-cappedBabbler and 10 home range surveys for Tickel'sBlueFlycatcher, both in disturbed and undisturbed habitats. It's a tough task to do scientific experiments to meet the standards of international journals. So we did our best to meet those standards, and now we are on the process of analyzing the data and publishing the findings. A huge credit should goes to the research team who worked hard in the field. I convey my heartiest gratitude to my supervisors, Professor Ben Goodell, Professor Christos Marmidas, Professor Ivo Jiang, Professor Sarat Kotagama, who always encourage me in every ups and downs in this PhD experiment. Without my research team, I'm nothing. Thus, I must thank to Mr. Harsha Atukorala, who hold the most of the weight of the research work, Mr. Indika Pabatua, Mr. Damindra Samarasinghe, Mr. Chandralal Kumara, Mr. Rukmal Ratnayaka, Mr. Tarandu Ranasinghe, and Mr. Umesh Fernando. At last but not least, I am grateful to the Conservation Food and Health Foundation of the United States of America for funding the project, the Special Talent Recruitment Grant of Guangxi University to Professor Ben Goodell, and the Chinese government scholarship for supporting me throughout the PhD project. I was delighted to have continuous encouragement from Professor Yuromi Manage Goodell, Professor Madhav Nikas Kumbura, Professor Devaga Veerkon, and Professor Inoka Kudavidanage. And I must thank the staff of Field Ornithology Group of Sri Lanka. And special thanks to Mr. John Rodrigo, Director Corporate Affairs of Hales Group at that time, and Mr. Udara Prematilaka from the head office of the Hales Group for coordinating with me to communicate with the plantation managers, the Halkol Estate Manager Mr. Amila Gannaru, Assistant Manager Anura, VO Estate Manager Mr. Asita Tilakaratna, and Assistant Manager Mr. Targa Jabadana, and Kundligama Thanapita Hena State Owner, and my father Mr. Dayananda for all the help provided during the project. Finally, I must thank the Sri Lankan Customs for facilitating the transmitter clearing process, and Mr. Janaka Pereira at the Telecommunication Regulatory Commission for helping me get the necessary approval from Customs and the Department of Import and Export Control. And I extend my gratitude to the Forest Conservation Department and the Department of Wildlife Conservation and Institute of Biology for granting me the necessary permission to carry out this work. Okay, hope you enjoy and gain some knowledge about our project, and hope to come back to you to talk more about findings and results of this project for conservation implications. Until that, bye for now from Wild FERN.